Today's video is going to be about the biosynthesis of integral membrane proteins. They're sometimes called proteins with transmembrane regions or transmembrane domains. And this process involves co-translational translocation, and in some cases is essentially identical to the first steps of the process that we talked about before COVID, the last lecture, and that's shown in this diagram, which is figure 1235 in your text. So that process was associated with the production of luminal ER soluble proteins, and we're going to come back to these in the context of vesicular trafficking, and they're going to be considered cargo at that point. But the idea here is that their synthesis, which coincides with translation, leads to the production of a soluble thing in the interior, first of the ER. Some mature ER proteins stay there, and we'll talk about how that can happen. But often this is an early step in the process of getting this thing as a soluble extracellular protein out to the extracellular surface. So I showed this slide in the previous set of lectures as an early introduction to co-translational translocation, but I did want to mention one thing that we didn't actually get to, and that is this step right here, in which the signal sequence that we talked about during the co-translational translocation is cleaved by a peptidase, signal peptidase, a protease, such that you are left with the signal sequence initially as an embedded short stretch of amino acids that eventually gets degraded. Some people claim that it actually has signaling properties. But in any case, the other product is the important thing as far as we're concerned, and that is this protein. Now, the book calls it mature. That's very premature, I would say, because this thing is going to get processed in a whole bunch of different ways. And I also don't like the fact that they don't distinguish this N-terminus from a regular N-terminus. This is a processed N-terminus. And in subsequent diagrams, I've indicated that by an N prime, just so that we'll know where a signal sequence has been. This is another slide that I showed that Thursday, was it? And we talked about GTP and SRP, SR. I don't want to go back there, but one thing that we didn't get to is the part right here that I wanted to focus on, and that is the formation of this loop, because that doesn't make any intuitive sense. So I want to talk about how that forms, and it's going to be important in understanding how transmembrane domains form. But the thing to keep in mind that's not indicated on this diagram, and I drew it in, is this positive charge, which is one of the things that makes this red, that causes this to be a signal sequence, that positive charge followed by hydrophobic residues. So what that's going to encounter at this stage, that also is not indicated, is a negative charge on the translocon, and I've got this written down in subsequent slides. But what's going to happen is the formation of a salt bridge during the very early stage of really the restarting of translation after translational arrest. That positive charge and that negative charge are going to interact in such a way as to form this loop. And that's going to happen in a number of cases for the rest of the day. And finally, there is another slide that I went over, and that's shown here, in which, although it's not entirely clear what's going on here, this is a bird's eye view looking up through the translocon from the luminal side. This is actually a side view. So these views don't coincide with these, so don't get confused by that. But we talked about the plug, that we're not going to talk about that again. But in terms of this pore, and especially this hinge, that's going to come into play a lot today. So what's going on in this cartoon version of the same thing is the idea that here's the pore through which co-translational translocation occurs. That's where the nascent chain goes. And there is a seam in the translocon. So rather than being a continuous donut through the membrane, it's a donut that's got a slice that goes all the way through into the donut hole and then almost all the way to the other end, and that is controlled by this hinge. So what happens is that the translocon can open up to the membrane, which is going to be sitting out here, meaning that the contents inside the pore can sample the phospholipid environment around the translocon, and that's going to be very important. First of all, to get the signal peptide out so that it can be cleaved, but it's also going to be involved in transmembrane domain synthesis. And here is a diagram that just indicates the relative proportions of these things. Okay, so let's move on to the actual business of making an integral membrane protein, again using essentially the same components that we talked about with the production of soluble, maybe secreted proteins. So in terms of making a membrane protein, it's necessary to identify 3 sequences that are needed in order to adapt the co-translational translocation machinery specifically to the synthesis of membrane proteins. The first one we already know, that's the signal sequence. And it's necessary to kick off co-translational translocation period. One additional one that we're going to need right off the bat is referred to as a stop transfer sequence. And a third is called different names by different people, but I like to call it a signal anchor for reasons that I hope will be obvious in just a second. So in terms of defining these sequences, it's nice to be kind of schematic, I think, right off the bat, and then we'll talk about how to use them mechanistically in just a minute. But if we want to compare these 3, then it's nice to think in terms of the signal sequence as a fairly low resolution schematic that might look like this, in which case we're at the N-terminus, the very N-terminus of the protein, however long the region after the signal sequence is, can be pretty much anything, sorry. But the signal sequence will be defined as a positive region, 1 or more amino acids at the very N-terminus, followed by a stretch of hydrophobic amino acid residues. And those are sometimes abbreviated HV, although I haven't used that abbreviation before, but just so that you're aware of it. That is sometimes the case. So that's our signal sequence. That's what it looks like. I'm going to introduce one additional component to it in order to compare it, especially with the signal anchor, which is going to look very similar otherwise. And that is that let's identify the region of the combination signal sequence nascent chain that is cleaved by a signal peptidase. So we could just put a caret there to indicate that this is the site that is going to be the new N-terminus, that so-called N' region, once signal peptidase does its thing. So if we go with that schematic, then the stop transfer sequence would be schematized sort of like this, and we'll keep it red just for kicks. But the idea is that you've got the N-terminus of the protein that is now going to be an integral membrane protein by virtue of having this thing in red. And if we want to keep everything roughly comparable, then the way that we would do it is like so. That is, we could put the hydrophobic region on the N-terminal side of the sequence, and we would put the positive charge on the C-terminal side of that sequence. So there's the hydrophobic residues here, and we can put this box anywhere, so there's no restriction, as we'll see eventually, in terms of where it is along the polypeptide chain. So we could stick it over here somewhere, we could stick it over here somewhere. All of those things are allowed in terms of the way it functions. And then finally, the third sequence that I want to identify is referred to as the signal anchor, and you can probably figure out what's going to happen here already. But if we, again, we go with the N-terminus, and this region again can be any length in the case of the signal anchor, Go with our red box to identify the sequence itself, and then finish off the protein. So now what's going to happen is that we're going to make a region roughly the same length as the signal sequence and the stop transfer, but in this case we have the positive charge on the N-terminal side and the hydrophobic region following it. In a way that's very reminiscent of the signal sequence, but there's no cleavage site here. So the difference between this and this is that, and essentially that's the only difference, other than the fact that the signal sequence has to be at the very N-terminus, and the signal anchor can be anywhere along this chain, and you see them all over the place. Collectively, these sequences are referred to as topogenic sequences, and nascent chain can have any combination of none, in which case it would be a soluble cytoplasmic protein, just the signal sequence, in which case it would be a soluble luminal extracellular protein, or combinations of two or more of any of these topogenic sequences, depending on what is necessary to construct the integral membrane protein.